Hello dear students. In the last lecture we have already started with the downstreaming process, and we have already seen few processes used in the product purifications, and we have also started with the product isolation, which we have already discussed that in this process the separation of the product is carried out by different methods like liquid extraction, adsorption, ultrafiltration, precipitation are some of the unit operational operations or the processes involved in the product isolation. Now we will see all these processes one by one, in which first one is your liquid extraction. Now it is a separation process that takes the advantage of the relative solubilities of solute in immiscible solvents. Now there are different types of immiscible solvents, and uh, solubility of the every solute is different for the different solutions or the solvents. So by taking the advantages of the solubility of the different solvents, the liquid or the two or more immiscible liquids can be separated by using liquid extractions. Solute is dissolved more readily and became more concentrated in the solvent, in which it has a higher solubility. Whenever any particular substance has the high solubility to any solvent, then it gets completely soluble in that solvent, and when it gets completely soluble, the concentration of the solution goes on increasing. A partial separation occurs when the number of solutes have the different relative solubilities in two solvents used. Whenever the two different immiscible solvents are used, and there is difference in the solubility of the solute, then uh, proper separation of the product does not takes place. And there are it is very important to check some of the properties of solvents so that it will not affect the product quality. Now here the solute is your product, and it is very important to maintain the product quality and quantity while the separation and purification process. So one should keep in mind that solvent should be non-toxic, it should be highly selective for the solvent or the product, then it should be inexpensive. If it will be if the solvent is expensive, then the cost of purification and ultimately the total manufacturing cost will increase, which will lead to the loss to the producer. And uh, the solvent should be immiscible with broth and should have a high distribution coefficient for the product. If the solvent will get miscible or dissolved in the broth, then the separation will get difficult, and product purification will not be carried out easily. So these criteria should be fulfilled while adding the solvent in the liquid extraction process. Next method of product isolation is your adsorption. Now adsorption is completely based on the surface phenomenon. Here your product or the particles get completely adsorbed on the surface of the adsorbent. It is the binding of molecule to the surface and different from the absorption. Now in the process of absorption, the material completely gets inside the absorbent, but opposite to it, in the case of adsorbent, the particles get attracted or stick only on the surface of the adsorbent. The binding to the surface is weak and reversible. The compounds containing chromogenic groups are usually strongly adsorbed on the activated carbon. The particles having different types of colors can be easily or strongly adsorbed on the activated carbon. Activated carbon is used as an adsorbent for the process of adsorption. Common adsorbent used are activated carbon, silica gel, alumina, because they present enormous surface area per unit weight. As the surface area present on the silica gel as well as activated carbon and alumina is larger, सरफेस एरिया लार्ज असल्यामुळे काय होतं, पार्टिकल्सला ॲडसॉर्ब होण्यासाठी जास्त स्पेस मिळते. सो जनरली these common adsorbents are used. Next process is your ultrafiltration. Now ultrafiltration is completely a pressure driven separation process. Here the pressure is applied for the separation of your product, and this pressure is usually between 0.1 to 1 mpm. Ultrafiltration is governed by the screening principle and dependent on the particle size. Now here the separation of your product and the other particle completely depends on the size of the particle. In the ultrafiltration membrane, different types of pores are present, which has the size range between 1 nanometer to 100 nanometer or 10 to 2000 Armstrong unit. Thus it allows the retention or the stoppage of the compound with the molecular weight of 300 to 5 lakh Dalton. मेम्ब्रेन अल्ट्राफिल्ट्रेशन मेम्ब्रेनवरती डिफरंट टाईपचे पोर्स प्रेझेंट असतात. आता ह्या पोर्सची साईज जी आहे ती 1 to 100 नॅनोमीटरपर्यंत व्हॅरी होऊ शकते. आता या पोर्समध्ये काय होतं की डिपेंडिंग ऑन द मॉलिक्युलर वेट 300 to 5 lakh डाल्टनपर्यंतचे पार्टिकल्स आपण काय करू शकतो, रिटेन करू शकतो किंवा स्टॉप करू शकतो. टिपिकली द प्रोसेस इज सुटेबल for retaining the biomolecules, bacteria, viruses, polymers, colloidal particles and sugar molecules. Now this ultrafiltration process is generally used to retain or to stop the or to trap the molecules, different types of biomolecules as well as bacteria and viruses or the different types of polymers, and it helps in the prevention of contamination of your product. Now here is a diagram of your ultrafiltration or the ultrafilters. Now upper side you can see the feed side is present, then your permit side, then your porous membrane support and the macromolecules. Now here you can see the upper white layer is your porous membrane through which your solution or the content is passed. On the feed side, the macromolecules, which are larger molecules, they remain on the filter or on the porous membrane, and the other particles are passed through it through the membrane support. On the down side, here you can see the ionic species and the water molecules, means the liquid content is passed through the porous membrane and the macromolecules or the larger molecules are retained on the surface of the membrane. Then the next process is of your precipitation. Now, formation of the solid in the solution during the chemical reaction. Now, precipitation, generally what happen in the liquid, there are different types of particles present. In the process of precipitation, at the bottom of the solution, the solid get gathered due to various chemical reaction, and the solids formed at the bottom are known as precipitate, and the liquid remaining above the solid is called as your supernatant. In this process, what we do, we separate the solid particles from the liquid. म्हणजे काय सॉलिड पार्टिकल्स बॉटमला collect होतो, that solid particle is known as your precipitate, and the remaining liquid is known as supernatant. In diagram also you can see the complete suspension was added after the adding different some chemicals depending on the nature of the solvent. The solid precipitate is formed at the bottom of the tube, and the upper liquid is supernatant remains above the precipitate. Now, different types of salts such as ammonium and the sodium sulfate are used for the proteins to precipitate. Now, here different types of chemicals, examples of chemicals or the substances are given which are responsible for the precipitation purpose. For the precipitation of proteins, ammonium or sodium sulfate is used. Organic solvents like methanol is used for the precipitation of dextrins. Then the chilled ethanol and acetone is used for protein precipitation. Non-ionic polymers such as polyethylene glycol is used for in the precipitation. So these different types of salts or the solutions or the solvents can be used for the precipitation of different depending on your product type. Now, these processes like filtration, precipitation, ultrafiltration may be there. These processes are done for the separation of your product. Till now, we have seen the process of product isolation, in which we have seen the different methods of precipitation, then ultrafiltration, adsorption, and liquid extraction. In the next lecture, we will see the methods of product purification. Up till now, we have seen the separation of insoluble particles and from the broth, then this isolation of your product, and in next lecture we will see the purification of the product.